Okay, so welcome everybody to Phylogenetic Trees Back to Basics, the Q&A session. Thanks very much for joining our follow-up session from around our 9 venues around the country. Today, once again, we're joined by Associate Professor Michael Charleston from the University of Tasmania, and he's worked on a few questions that have been submitted since our last session together. We're also joined around Australia by a whole host of wonderful facilitators who have volunteered to help out their venues and bring training into their local areas. Today we're also joined by Gareth Price, who is standing by to answer any tricky Galaxy Australia questions if they arise. Just a reminder of how we're going to interact today. Everyone else other than the speaker will be in mute, but we encourage you to ask questions and interact as much as you like with your local facilitators. They can contact us via the online chat window within the Zoom meeting, but you can also freely add all of your questions and comments to the discussion board. That's live now, so please head there and we can converse that way with all the facilitators, participants, and also our lead trainer. The session will be recorded as the Part A was recorded, and that's Part A is now available on the Australian Biocommons YouTube channel, as will this one. We'll let you know when this session's being uploaded there. So anyway, now this session today is going to be an informal Q&A session, and I'm going to hand straight over to Michael Charleston to answer the questions that have come in.
I got a few interesting questions. There was a fairly common theme through these questions, I guess that I should talk a little bit about that common theme before I continue on with the specifics. So that was that a lot of questions are relating different, potentially different trees estimated from different loci, like different genes or maybe different parts of a chromosome or maybe nuclear versus mitochondrial DNA, and how to accommodate the excuse me, how to accommodate the differences between the inferred trees that you might get from those different data sources. So this in general is something that you need to be fully aware that then you might have different loci, like different genes or perhaps nuclear versus mitochondrial DNA, or different gene copies from a gene family, that you might get the tree completely right for that locus. That is, you've got the divergence patterns absolutely perfect, you've got your tree right, but they might not agree, and they might not agree with the species tree, which is, if that's what you're after, then it might be a bit confusing. So there are ways around that, and we'll cover those as well.
Can I just interrupt there for a second, Michael? Sorry. We've had a query about whether you can zoom in on the text there, but I'll also let participants know that these words have been shared via, if you go to that schedule document, there's a link there where you can view this text on your own screens as well.
So the first one's from Sandip Kamath at JCU. The question is, I work with protein sequences from invertebrates, crustaceans and mollusks, and that's data from GenBank and UniProt. Questions are around which algorithm to use, neighbor joining, UPGMA or maximum parsimony, and why, and what's the difference. So I guess I'll answer that one first, and then I'll come back to the other questions. So on which algorithm to use, first of all, definitely avoid using UPGMA. That's unweighted pair group method with arithmetic mean. It's a terrible, terrible program or terrible algorithm. The reason that it's terrible is that it's extremely sensitive to deviations from what we like to think of as the molecular clock. And that idea is that the distance between the root and the tips is more or less the same, or it's close to being the same for all of the taxa in your group of taxa that you're interested in. So definitely avoid that. And no one presenting a UPGMA tree should be taken seriously. Point and laugh. It's a terrible method. Neighbor joining gets around the problem that UPGMA has, so that if the distances on the in your group of species that you're interested in, if the distances are tree-like, if there is a tree that fits those distances, so that the distance between each pair of taxa is the same as the path length on that tree between those taxa, then neighbor joining will definitely find you that tree. So in a sense, it's the perfect method if you have tree-like distances. Now, if you have other sorts of data like aligned sequences, then you can use character-based methods like maximum parsimony, which we didn't discuss much, and maximum likelihood, which we did discuss a bit more. And if you've got aligned sequences with characters like DNA sequences or codons or amino acids, then maximum parsimony and maximum likelihood are both good ones to use. Another a factor when you're choosing your methods, and so far I've said use them all except UPGMA, is the size of your data set. So if you have a really huge data set, like thousands of taxa, then maximum parsimony and maximum likelihood are just too slow, and they won't run, and they won't finish by the time your grant funding runs out. So I would then start with the neighbor joining. And just remember that one of the things we did first in the workshop was build a neighbor net, which is a phylogenetic network, and it's the default output of splits tree. And that was a really great way of looking at your data first, so that you could see where there are some regions perhaps of the tree that were well supported without much conflict, and some other regions might have more conflict. So I would advocate using neighbor joining and maximum parsimony and likelihood, and you really want them all to roughly agree. Of those, probably maximum likelihood, if those trees do disagree, maximum likelihood is likely to be your best, most reliable, and it's also the most statistically defensible tool to infer a tree. So in case of disagreement, probably go with the maximum likelihood tree. And again, do start by looking at a neighbor net or some other kind of sensible phylogenetic network. Okay, so the next question is how to deal with protein sequences from a database when you have partial sequences versus full-length sequences and different isoforms of the same protein. So this is a different kind of a question. So my response to that would be, this is on the next page, I hope you can read this text. If you have partial sequences, then you should align them as well as you can anyway, as well as you would complete sequences. And you need to hope that you've got enough loci that you can align it, and so that you can find homologous sites. If you've got enough homologous sites so you can resolve that phylogeny, then you'll be fine. But if you don't get enough overlap of those sequences, if you're missing a lot of sequence information across your taxon set, then you need to find some more data, basically. Either put maybe look at the coding sequence itself, the DNA, or you can look at some other loci. Maybe even look at morphology or something else to resolve that, because you simply don't have enough data. So partial sequences can be a real problem if it's very, very patchy. If you simply don't have enough information, then throwing them in willy-nilly, even if you've got a nice bit of sequence from this taxon, another nice bit of sequence from that taxon, but if you can't find where they align in your with the rest of the sequences, then they're useless, and you should just either get some more data or drop those taxa. This is a bit of foreshadowing for one of the other questions, was what are the common flaws that I find in phylogenetics, and one of them would be and is keeping data when they're really unhelpful to you. You don't need to include them if it's just not enough to go on. The second part of that question, which is about isoforms, so different isoforms of the same protein, is very interesting, and I haven't really thought about it very much. But if your if your isoforms have come about due to, say, genetic variation and or modifications after transcription, then that's quite easy. You just include all the sequences of the isoforms, and then you hope that they will all group together in the tree. In fact, you can force them to group together in your tree. For example, in IQ tree, and there's a link further down showing you how to do that. So you can require, if you know that they are isoforms of the same protein in the same species or in closely related species, then you can force them to come together in the tree. On the other hand, you might have isoforms that are due to alternative splicing. And if that's the case, then I think what you should be doing is trying to identify which sections of the amino acid sequence correspond to which exons and align those. So I've got a little sort of toy example here I'm just highlighting. Suppose you've got, say, exons E1 to E4 and a couple of sequences. There'd be protein sequences with E1, E3, E4, and then E1, E2, E3, E4. Clearly, you're missing E2 from the first sequence, and you would expect your alignment to find the homologous exon, product of the exons, the amino acid subsequences, lining up in that way. If they don't, then, well, you know your protein better than I do. You should probably force them to align that way and deal with the fact that essentially what you have is, in this situation, you've got 4 different loci, and it's similar to the whole gene tree, species tree problem that we'll be covering probably quite a lot today, where you've got some sequences simply missing information at a locus. In this case, it's an exon. In other situations, it might be a gene. And you model them and build phylogenies on them separately. So you'll build a phylogeny on E1, you'll build a phylogeny on E2, you'll build a phylogeny on E3, possibly E3 and E4 together, but certainly you wouldn't do them all in one go. So in theory, that should be fine. If you've got alternative splice variants, then you should, and you've sampled those variants from the same species, you can fix them together so that they're going to be constrained to be in the same part of the tree. They're going to be each other's closest relatives. And that's quite easy to do. The format for that is simply the same format as how you would describe a complete tree. It doesn't need to be complete. You can just group together sets of taxa that you think should go together. A lot of phylogeny inference programs can do that. We used IQtree the other day, and you'll be pleased to know that IQtree does that as well. I've just put a little link in here. So iqtree.org/doc/advanced tutorial, and that advanced tutorial has a section on the page, constrained tree search. It's very simple to do. So that's my answer to that bit. The last one is Mega or GelView, also a good option in comparison to Galaxy and SplitsTree. I confess I don't use GelView, and I have used Mega in the past, but I prefer using SplitsTree and IQtree for my tree finding. SplitsTree is great because it starts with that network view. And Galaxy is, as you know, of course, is a hosting platform for a whole lot of tools, and IQtree is one of them. And so I'm very happy with IQtree. We did consider using Mega for the workshop, but we decided against it because, in fact, the current version of Mega isn't identical on both platforms, common platforms, Mac and Windows. So it wasn't going to give everybody exactly the same experience, whereas doing it on Galaxy was. Note, of course, you can download IQTree and you can run IQTree on your own machine, and then it's fine, and will run just as well as it does on Galaxy, depending on the amount of compute power you have on your own machine or a server. Then you can make it go pretty fast. So I don't really have a clear answer on that one, Mega versus GelView, or GelView versus the others. But as I said, my favorites to go to are SplitsTree and IQTree. When we do a discussion on MCMC and Bayesian methods for phylogenetics, which we are talking about having sort of early-ish next year, then we'll talk about using BEAST, which is another absolutely standard tool in the phylogenetics toolkit. And that doesn't run on Galaxy as far as I know. Maybe it will by then, I don't know. That takes an awful lot of compute, but we'll discuss that later on, offline, when we're planning. Okay, so I didn't really have more of an answer to part 3, really because I don't have a firm view on whichever tool you want to use. Ultimately, it's use a tool that's comfortable, and what matters is the algorithm and the optimization criterion that you use more than the software. The software of these ones is pretty well tested. Mega has huge numbers of users. SplitsTree, IQTree have huge numbers of users, and they're very well tested and they're pretty reliable. So it's really just down to user experience. Okay, so this is a question about multiple crop and insect genomes. So yes, I understand you're facing several challenges, particularly with crops and with insects. And so the following questions are: how can one conclude from a phylogenetic analysis that specific gene families have been expanded or contracted, reduced or lost? If, for example, in an MSA, that would be multiple sequence alignment, redundant sequences are removed. Well, so that's that 1A question. Well, there's a bit to answer about that. First of all, so here we are, genes, specific gene families. The reason, I guess, we should start with why you might have, the alignment program will have removed those redundant sequences, is they're not giving you anything new. They're complete duplicates. Because they're identical, all of the phylogenetic methods are simply going to put them into the same clade anyway, with a zero length branches joining them together. So they're pointless to include. So, and all they do is basically muddy the waters, make extra compute required. Some tools, including IQTree, I think, off the top of my head, will also remove duplicate sequences if they are detected, and then just put them in afterwards, because it doesn't help anything. So that's why they would be removed. I think then if you've got a situation like that, and you've got duplicate sequences, then you should simply add them straight back in afterwards. You can notice that you've got, say, 5 identical sequences at one leaf, and you might then be able to conclude that if you've got 5 identical sequences at a similar leaf, perhaps that clade has duplicated some gene or some genes in that clade. But if they're duplicates, you can't necessarily, if they're perfect duplicates, you can't necessarily tell that this is an expanded or contracted gene family. It might simply be you've got more samples. So unless I'm getting the question wrong, I think it's almost not a phylogenetics question, I suppose. Now, one thing that you could do potentially is, if you have a lot of sequences that aren't perhaps identical, is build a tree on the whole lot. And if you find that you have multiple subtrees that are matching a clade, so matching a species clade, and multiple parts of your gene phylogeny are matching that clade, then it looks like there might have been a gene duplication, or perhaps a set of genes have duplicated at the root of that clade. So that would mean that perhaps you've got multiple copies of genes and you've sampled them all, and that you might be able to use to conclude that you've got an increase in the size of gene family at the root of that clade. On the other hand, if you haven't got all of those gene copies sampled, what will happen is you'll end up with a gene tree that doesn't look like the species tree. We didn't really talk about that much last time, but one of the things that can happen is, even if you have the gene tree exactly right, some processes exist which mean that the gene tree doesn't match the species tree. And these processes are things like gene duplications, in which case you have multiple copies of the gene going down to the leaves of the tree, the extant taxa. What's called incomplete lineage sorting, which is where after a speciation event, it's not the case that both new genes, new copies of the gene, go down both lineages. If you don't need both copies for function, then it's not necessary that both lineages will contain both copies. And so that's a loss. It's called an incomplete lineage sorting. And that's a way in which the gene tree won't look like the species tree. Another one, and this is, I think, cropped up a couple of times in the questions, is that there might be a lateral gene transfer or horizontal gene transfer. And that's where genes can go from one lineage across the tree to another lineage. Perhaps this happens a lot in bacteria, not so much in eukaryotes, where then you have a disagreement again in the shape of the gene tree, because it's now got to jump across from one lineage to another, and it no longer fits the species tree. So there are a lot of these different ways. And another one, I guess, to finish off would be simply that you don't find, or you don't sample every copy of some duplicate genes. And that will give rise to a disagreement, particularly after duplication has happened. If you simply don't get all of the genes that are homologous, that all have the same evolutionary history as the species, then you'll may well get the gene phylogeny right, but it won't match the species phylogeny. So it's quite tricky to do. I think if you're trying to infer gene family expansion and contraction from a tree, then you kind of need those sequences to be different. And it's worth building the tree on the complete set, including adding back those duplicates if you really need them, and then comparing that to what you think might be going on with the species tree. If you don't have a species tree, then what you're looking for in the gene tree or gene family tree are repeated patterns of divergence. For example, one subtree looks just like another subtree, and with the same estimated divergence times, you would conclude that, or you might conclude that they have diverged together because they have duplicated a bit earlier in time. You might find the root of those two subtrees corresponds to a speciation event in your species tree. I hope that's clear. If it's not clear to the person who asked that, Paul, then flick me an email and we can discuss it further. There's a lot of information in the literature about this issue, in particular of gene tree versus species tree, and inferring species trees from gene trees is difficult but doable. Okay, next question is how you would infer events such as a whole genome duplication in the case of polyploid genomes. Well, I think if you know it's a polyploid genome, then you know there's been a whole genome duplication at some point. I guess it's a question of finding out when it was. So basically to do that, you need enough copies from both descendants of such an event, WGD, whole genome duplication event, so that you can make phylogenies based on the complete set. And you would hope that those phylogenies will look the same as each other. You put it all into the same analysis, and you should end up with two subtrees in your big tree, which look like each other, more or less. If they're identical, then you're very happy. And if they're not identical, then it's possible that you've either had some sampling error, that is, you've missed genes. You might have some pseudogenes kicking around that have lost function and might be hard to sequence and might be hard to recognize. If there's some lateral gene transfer going on, then you'll get some discrepancies. But if they match perfectly, you're laughing. If you're doing, say, shotgun sequencing, you're doing the whole genome sequencing rather than focusing on a particular gene, then I think it's that will depend on whether you're actually aligning it to a reference or doing de novo genome assembly. Again, if that's the situation that you're in, trying to identify whole genome duplications based on massive throughput sequencing or next-gen sequencing, and you don't have a reference, then that's a whole lot harder. And we'll probably have to discuss that one online too. This is a very hard question. So these are all methods, I think, in these first two questions are really touching on the relationship between gene trees and species trees, or the evolutionary dynamics of different loci and how they relate to each other and to the species tree. And in that realm, which is usually called co-phylogenetics, there's a whole lot of methods based on mapping the locus trees, so gene tree or other kind of locus, into a species tree. And so that might be mapping with what's called a duplication and loss model, or DL model, or a duplication and transfer and loss model, DTL model. That's the one that allows horizontal transfers or lateral gene transfer events. And these methods exist. If with a duplication loss model, DL, you can map the species tree into the gene tree into a species tree very easily. They're fast, they're efficient, and you'll get an optimal answer. DTL, the problem is these horizontal transfers. These make everything much, much harder. And you can, while you still can map the gene tree into a species tree, you can't guarantee, in the general case, that you'll have an optimal solution. You can't always tell. There are plenty of methods for this, and these methods either usually minimize some kind of total cost. If you might remember from the other day, we spoke about edit cost for alignments. It's a bit like that. It's simply adding up a weighted sum of the sorts of events that you need to include, like losses, co-speciation events, which don't cost anything usually, horizontal transfers, and duplications. And that can give you a sort of a total cost on one of these maps of the locus tree, gene tree into the species tree. So those do exist, those work quite well. And if you have gene trees and a species tree, then mapping the gene tree into that species tree can be a really good way of telling whether you've got a duplication event going on there. Not necessarily a whole genome, but it applies also to whole genome. It also applies to more restricted problems. Pause for breath. The next part of the question, which is to see, when is it preferable to use protein versus DNA sequences and why? That's a good question. So this is what I've got as number 3 down here. Ideally, I would say use both if you have both. Use all the data you have unless it's terrible, useless data. So, but there's an awful lot more that you can do with DNA than you can do with protein sequences. And the reason for that is that we have a whole lot of mathematics and wonderful tools that can be used to recognize specific evolutionary dynamics of a taxon set using DNA, but oddly can't be used in the same way for protein models, say 20-state amino acids or 64-state codon models. And the reason for that is that these DNA models are much simpler. They have fewer parameters in them. In a substitution matrix, which gives you the probability or the rate of changing from, say, one state to another, like an A to a G or an A to a T, there's only a few parameters in these models. You'll probably have heard of the GTR model, General Time Reversible model, that only has 9 tunable parameters, 9 parameters that can vary from, say, locus to locus or from gene to gene. 9 might seem a lot, but it's actually not too bad, particularly if you've got an awful lot of data, say 100 taxa, 5,000 loci, then maybe you can probably infer the values of those parameters really well because you've got a lot of information. And that means that you can get a well-fit substitution model for your data set, and it means that you have a really good chance of getting the tree right. On the other hand, the protein models, and say the amino acid models or the codon models, they have a heck of a lot more parameters. So for a 20-state amino acid model, you could conceivably have about 100 parameters or 200. That's about 200 parameters. And that's an awful lot. And it's much harder to estimate the value of those parameters in your transition or substitution model with the same amount of information. And in fact, if you're converting from DNA down to amino acid, you have less information. So it makes it really hard to tune those models to get the best set of parameters for your data set using an amino acid or a codon model. So instead, what you have to do if you're using amino acids or codons is you have to pick a good model based on, for example, its current popularity, the database upon which it was based. There's WAG models, there's JTT models. Those of you who are interested in these kind of data for phylogenetics, you will have come across those models. There's Blossom, a really old one. I think I showed you in the presentation the other day. But you have to pick one and then use that one for your analysis. So it means that it's not possible to best fit that model to your data. You're kind of stuck with what you get. Having said all of that, so which is essentially saying that the DNA is a better choice when you're doing taxonomy and phylogenetics. Having said all of that, though, you might find that you're after some divergence time for a really anciently derived taxonomic group. So you're after something that evolves very, very slowly. And protein sequences can be more conserved than DNA sequences. So if you have that situation, you might actually still want to use protein sequences, even with this disadvantage that you can't fit the substitution matrix to your data. You just have to pick one. Protein sequences can be extremely well conserved, though. So if that's the situation, if you're trying to answer a question about the systematics or the phylogenetics of a really old clade, then that might be a valid way to do it. Just as a final note on that, I wouldn't recommend converting DNA to protein sequences. If you have DNA and you're wondering whether you want to just use amino acid sequences instead, because the models that deal with the coding sequences can be much, can employ different partitions, because you can split up your data for quickly evolving sites anyway. So first, second, third position sites anyway. And so that variation, like the codon models will capture that, but the DNA models will also do that anyway, and you can tune them better. So I just wouldn't recommend going to protein sequences, amino acids, or treating them as codons. You might find amino acids as more conserved, but you might not. I just wouldn't go there. All right, so then on to question D, which was how do you infer HGTs? How do you infer horizontal gene transfer events? There's a lot of ways of doing this. So here we are, inferring HGT. It's actually very hard to do. It's a computationally complex problem to try and solve. The way that you do it is by constructing trees or inferring your trees from a set of genes of interest and showing that the best fit trees are statistically significantly different from each other and or from the species tree if you've got one. So just to repeat, to know that HGT is going on, you have to show that the two trees, you have to show that two trees, either two genes or more, or a gene and the species tree, are statistically significantly different. There are ways to do that, and one way, the first one would be to say build trees from your different loci first. I've got genes down here. It could be genes, it could be exons, it could be spacers, whatever. And then see whether the tree that you get from concatenating those two sets of sequences is worse. Okay, so if it's very different when you concatenate the two sequences, worse than either of them, say it has a much worse likelihood score or a much bigger parsimony score, then if that's the case, then you can say, well, there probably is evidence that the trees are different. When you've got evidence that the trees are different, then you can compare them properly under the assumption, the safe assumption, that they actually are not just from the same evolutionary history. And you might simply have got them by chance a little bit different. So if they're significantly different, then you can compare them under the assumption that they have different evolutionary dynamics. So that's one way. Another way is to use a sliding window. So a sliding window is if you can imagine, you've got your alignment and you move a sliding window along it, and at each position of that sliding window, you sort of build a tree or look at the phylogenetic signal that you have in that sliding window as you move it along, and then you look for changes. And for example, if the sliding window over here says one tree and over here says it's a different tree, then it looks like you've got a change in evolutionary signal, in phylogenetic information between the positions of those two windows. So somewhere down the middle of them, two sliding windows like that. And there's been quite a lot of quite a lot of work that's been done on identifying those sorts of changeover points, those cutoff points, because it's exactly the same idea as almost exactly the same idea as recombination event. And so James McInerney has done a lot of work on this. You can just Google him. I don't have a link in here, I'm afraid. And he has methods that are that are designed to find these recombination points, which will work perfectly well for also identifying in a concatenated alignment when you've got a change in phylogenetic information. Another option might be looking at something called partition finder, which is created by Rob Lanphier and others, I think, or concordance factor analysis. So partition finder will seek to find the partitions of your data. That will be sets of sites. For example, a region of your gene from 1 to 500 or from 501 to 600 or something like that, that have well-defined and different evolutionary dynamics. And you could look at those partitions that partition finder gives you and build trees on those. Again, see if there's some HGT going on because you have significantly different trees. Another approach is called concordance factor analysis, and that is, I've just put a little link in here, and that's a similar sort of process where you are looking along an alignment to see at what point does the concordance, the agreement between your sequences around a particular point, when does that concordance change? You'll be pleased to know that is built into IQ-Tree as well. So if you've been using IQ-Tree, then that's a fairly straightforward addition to your analysis to run concordance factor on there, and there's a link there. Okay, so there's a whole lot of these methods that are built around relating locus trees to species trees. So gene tree, species tree, it's known as the gene tree species tree problem. And for HGT, you do need to use one of these DTL, duplication transfer loss models. There's a few good programs. So Ran Liebeskind Hadas has written a program with his students called Jane, and it's pretty good. It's well maintained. There's another program called Tarzan. You might be wondering what about the names. I'll tell you in a sec. And that hasn't been updated, I think, since 2005, so it might be abandonware now. Pierre Legendre wrote Parafit, and that's actually a method that's in some R packages. And just to see if I can, I'll put it into here. So R packages. I'm just putting it into the discussion board. Ape and, don't capitalize that please, Ape and another one called Fangorn. Oops, that's not how you spell Fangorn. Good packages for phylogenetics in R. If you're interested in using R for your analyses, which probably a lot of you are. I've written a program a long time ago, and it's not actually abandoned, but it's not well maintained. It's called TreeMap. It can be used for visualizations. And the more people that hassle me to update it, the better, because then I'll get onto it. So HGT is, in summary, you find that by comparing trees with preferably with a species tree, and you can then map the gene tree into that species tree and see whether host switches have taken place. You should do that once you know, or sorry, horizontal transfer events, and you should know whether know in advance that the two trees are significantly different before you try. Otherwise, you're essentially just pretending that it's there when it might not be. I promised an origin of the names, Jane and Tarzan, and there's even another one called Mowgli from Rudyard Kipling's The Jungle Book. And the reason for that is that I named a structure a jungle a long time ago, which was meant to tackle this problem. And if you want to cite my jungles paper, go for it, but it's maths. So hence Tarzan and Jane came along. It's a shame because I wanted to use Tarzan. Anyway, right, next part, number 5. Gosh, there's a lot to this question. Based on inferences 2A, 2B, 2C, 2D, and 2E above, 2E is not above, how would you design your sequencing/sampling strategy to ensure you have sufficient data to confidently draw these conclusions across and within taxa, that is? Well, that's a very, very hard question. I think it depends on a bunch of factors. Obviously, we want as much data as we possibly can get. And you do want to have informative data, phylogenetically informative data. So to start with, you want to have well-balanced taxon sampling. There was a study many, many years ago from David Hillis and John Hulsenbeck. It was either a Nature or a Science paper, And that was talking about the effect, the beneficial effect of adding taxa to phylogenetic analysis, so adding species or genes or strains, in order to make the branch lengths on the resulting tree comparable. So I've said comparable there. I sort of mean within a factor of 10 of each other. It's then and since been confirmed, it was then shown and been confirmed many, many times, that if you have branch lengths in the true phylogeny that are extremely different from each other, say a couple of orders of magnitude different from each other, then it's very likely to cause problems, because those short branches get swamped by the information coming from the long branches. There's a factor I think I discussed last time called long branch attraction, and the long branch attraction will draw those long branches relative to the short ones together just because they've had a long time to match by chance. If you have long branches and very short branches, this is exacerbated. So if the short branches are very tiny with respect to those long ones, then you can easily get those inferences wrong. So you want to avoid that if possible. So if you can break up the long branches by perhaps subsequently adding more data, more sequencing, then that's a good idea. Although, having said all that, you also might have some other constraints. So you might have you might be constrained by what, depending on what sequencing you're doing. If you're doing amplicon sequencing, if you've got particular genes that you can sequence because you want to fit them in with existing analyses, like you've got 16S, you can get 18S, you've got CO1, then that will constrain what you can do. It will constrain what sort of age you can work with, what divergence times of your clades you can infer by how fast they're evolving. So if they're evolving very, very slowly, you certainly can't use them for subspecies resolution. If they're evolving very, very fast, you can't use them to infer roots of the tree of life, or deep branches in the tree of life. So that's a factor. So it'll depend on what you're working on. It'll also depend on how speciose that taxonomic group is, so how many species you have in a clade. So how ancient it is, whether it's coding or not, whether it's nuclear or mitochondrial DNA, how expensive it is to get those, lots and lots of issues. And further to that, if you, for example, want to pick up specific events like HGTs or whole genome duplications, where have we got whole genome duplications way up there, then you probably need very reliable trees for all of the loci that you're interested in. Where am I? Back to number 5. You don't want it patchy. You want very, very good coverage, in the sense that for each sequence you're including, you want all of the loci to match up across, because you're after disagreement between the phylogenetic signal which is given to you from those loci. So HGTs are hard to estimate as it is, and they're much harder to estimate if you're missing data. So you really want good, I would say good coverage. You don't necessarily need duplicate sequences from each species or each taxon, but you want at least one good sequence from each taxon of the loci that you're after. I'm not sure whether that really answers your question. Let me go back and have a look at it again. Across and within taxa to draw these conclusions, infer events, So it's not really about C. I think we've covered that. That's part C is you should probably use DNA. Looking at specific gene families, if you think there's some gene duplication going on or genome duplication going on, you need to make sure that you get probably whole genome sequence, so that you know how big your gene family is, or if you've got a whole genome duplication event happening. I assume you can karyotype it looking at it under a microscope and count chromosomes and stuff. All right, so I'm not sure what to add to that. And if you have further questions, I would say get in touch with me by email, and we can see if we can get some more detail back to you on that. It's a common question, but it always depends on the what you're trying to get out of your analysis, your budget, the tax are involved, and so on. Okay, so that's part way through Paul's question. But now we're on to, I guess, one of my favorites of the day, apart from gene tree species tree, which is a research interest of mine. But the favorite one of the day, I think, is what are the common flaws you have observed in phylogenetic analysis? And I think that there's a big list. I haven't actually finished all of this list, but I guess they fall into a couple of simple categories. Part one, not looking at the alignment itself. This bothers me immensely. People who have and this is a serious problem. If you have a massive amount of data, you have a workflow, and you run it through your workflow, and you don't look at the steps along the way. So that could be you have shotgun sequences, you have you've assembled de novo from your bugs of interest. You don't have a reference, and you make contigs, and you put them without looking at them through an alignment program, and you put those alignments through some trimming, and then you put the trimmed alignments into a phylogenetic analysis, and you look at the end and you go, great, there's my tree. Not looking back at the alignment is always worrying. So please always do that. There's also, I guess another issue is a big one is, and you don't see it too often in publications, but you do see it occasionally, it's just fixing on one method. You don't need to do that. Most of these methods will run quite quickly. The things that take a long time are Bayesian analysis using something like Beast. Even though Beast is tremendously efficient, it does a lot of work to give you an output. IQ tree runs very fast, and it's optimized to do its thing very, very quickly. Another one that works extremely well is RaxML, and it's also on Galaxy. They both work super fast, so there's no reason to not use multiple methods. As I say, one of my usual ports of call when I'm doing a phylogeny myself is to start by looking at the splits tree output, a neighbor net. The reason you should look at multiple methods and not just fix on one is that you have, not only that you have time, but also you expect the phylogenetic signal to be robust to whatever arbitrary choice you make on which approach is best. You want the data to actually tell you the story, even if you get your model wrong or your method wrong. And if you get your method, well, if you get your model wrong, then you're just one of the rest of us, because the model probably is wrong. And so assuming that it's always definitely evolving in a particular way, or that neighbor joining is the best way of doing it, is missing the point. You actually want the data to give you the tree, regardless of whether you've perhaps picked the wrong method to do it. If they are different, then you go back and you look at where they're different, and you might go back and look at the alignment again to see if you made a mistake there, or you look at your splits tree output, the neighbor net, and see if the two methods or more methods are giving you potentially different resolutions of that neighbor net. And if that's the case, then maybe it's okay. Alternatively, it might be that one method is picking up one tree, another method is picking up another tree, and you've actually got a horizontal gene transfer event going on. So you really, really do need to look at multiple methods. And and that your decision about which method to use to not matter. Another one that I hate, common flaws, this is a list of bugbears, I guess, is assuming that Bootstrap values are confidence intervals, and they're just not. What they are is they are measures of the robustness of the signal that you get when you're resampling the data. And that robustness, if you like, that Bootstrap value goes up when you have more data. So I was at the Australasian Evolution Society conference last week, and there was a great talk there by Rob Lanphier, and he was raising this exact issue, saying that the reason that we get more and more confident in our trees is that we're putting more and more data into them. That is, more and more sites, more loci. And a natural consequence of the way that that bootstrapping process goes is that the Bootstrap value goes up and up. It doesn't mean that your tree is right. There can still be systematic errors. You might have got your models wrong, your method wrong, bearing in mind my comments for the previous point. And so Bootstrap values don't mean that you're confident in your branch. At least they shouldn't mean that. Point 4, not thinking about or not understanding methodology, which is related to using point 5, using the default parameters only, is really not something that we should be admitting happens. When you're doing a phylogenetic analysis, you should know how it's working. I've seen a few student presentations and talks and occasionally papers that make me a little bit suspicious that the authors actually haven't understood what the methodology is doing. It doesn't require a huge, deep understanding, but you should probably know what these methods do. Like parsimony minimizes total number of character state changes. Likelihood needs a probabilistic model, is statistically defensible, and roughly how they're working. It shouldn't be a black box. Point 6, I've done 4 and 5 together. Point 6, one problem I've seen many times, and one quite recently we'll have to redo a tree for, including data that are useless. The mentality being that I've got my data, I took a lot of money to get the data, I've got some I've got some parts of my sequence from that particular taxon, and I should include it. Well, if you can't align it, then it's not helping you, and it's probably making things worse. And if you've got some a sequence that is, or a taxon that is missing a lot of information, you may as well leave it out and say, look, we tried, it didn't work, and just accept it, that it's not going to help. It's possibly making things worse. Another kind of data that can get included that's useless is those parts of the alignment that are very tricky to align because they have a lot of variation in them. They may be very fast evolving, may have some insertion and deletion events going on that could go equally well, this part of the alignment or that part of alignment. And there are methods, for example, like G-blocks. So G-blocks, I'll just make a note to myself there, is a program that can help you filter out those problematic sites. So you shouldn't be emotionally attached to your data. I know sometimes it's very expensive and it's kind of disappointing when you can't use some data, but it's possible that data that are very gappy or very unreliable, lots of N's in your sequence, they're just not helping, and you should just admit defeat and get rid of them. Next common flaw would be assuming that there has to be a single tree. Trees are great, they're beautiful. You look at a tree and you think, this is fantastic, I have got my answer. It might actually not be the case that you have a single tree. For the reasons that we've sort of already discussed, there might be different trees that correspond to different loci that you've sequenced. It might be different genes, there might be nuclear and mitochondrial DNA, It might be, in fact, that you've got some hybridization, some full species hybridization going on. And so it isn't actually a tree. So don't assume that it has to be a single tree. Don't force it. Again, it's just generally good practice to start off with a network and then see when your network looks tree-like and see where in your data there might be some conflicting signal. And there are those methods I alluded to before, and concordance factor and the recombination seeking software that can help you identify where the information for a particular tree changes from maybe one tree to another tree. So it doesn't have to be a single tree. This is another one that I came across the other day, and this is number 8, using an overgeneral model across the entire alignment without allowing for perhaps different models in different parts of the alignment. And the reason for that is, I guess it's a little bit subtle, but it's an important one. So I'll give you a bit of a case study. I was working on an alignment last week, a phylogenetic analysis last week, and we had 1,400 or 400, some large number of loci. And the initial analysis had been done using the GTR, general time reversible, plus I. So I is a certain proportion of the sites are invarying, they can't change, plus gamma, and the gamma part means that some sites are evolving faster than others. So we're accommodating rates, variable rates of evolution across sites. So the GTR plus I plus gamma. It's a pretty general model, and it's the most general model that people tend to use. And so, no surprise, if you try and fit a model, find the best fit model for a massive data set like that, it will be the best fit model. But the interesting thing was that the if we found the best fit model for the individual loci, none of those best fit models was GTR plus I plus gamma. They all were perfectly happy with a simpler model. And the reason that it was fitting to a GTR plus I plus gamma for the whole alignment is that there was a whole lot going on, and that was the most general one that could be attempted to be fit. It just happened to be not one that fit any of the individual loci. So it's definitely worth breaking those partitions down so that you can find a model that is general enough, but not too general, so that you can still use it to infer a tree. The next question, I've got something to correspond to, is from Eva, and the question goes: I work on recombinogenic bacteria, and I would like to know solutions how this issue can be addressed in phylogenetic trees. I'm actually not sure what the issue is. If it's finding recombination events, then we've sort of discussed that a little bit. And that software I mentioned by James is pretty good for that. It's designed for that. He's been working on it for years. So it's a well-solved problem, I think. If you mean recombinogenic bacteria producing recombinant DNA, I'm not sure what that is to do with. So if you have perhaps a clarification that you can put in now, that would be great. And if not, then I think we'll have to leave that because I'm not sure what you're getting at there. Okay, so next one is from here at UTAS. It's Adam. And the question goes: when dealing with a small number of OTUs, about 10, so OTU, operational taxonomic unit, taxa, and relatively short, about 600 base pair coding sequence, is there a particular method of analysis you'd recommend over and above an NJ tree? The project is looking at geographical separation within an invertebrate species. So absolutely, yes. I wouldn't, I would build a neighbor joining tree, but I would be using maximum likelihood because this is a tiny data set. It's really easy to do an exhaustive or branch and bound, which is implicitly exhaustive search for your best tree using maximum likelihood. And it wouldn't even take long to run. It's tiny data. And Adam, if you, since you're here in Tas, if you want some help with that, I'll help you out there. So I would say, if it's, and also because it's coding, then you know that the first and second codon positions are going to be evolving a bit more slowly than the third codon position, which has got that 2 or 4-fold redundancy in it. Assuming you know that where the open reading frame starts, then you can use models that account for that third position redundancy. IQ-tree can also handle this, which is nice. And so here's a little sort of a sample data set that you can, that shows the format that you would need to partition your data in IQ-tree. I think, maybe that's not the right one to do, actually. So now I think about that, I'm going to go elsewhere and pick up that link, so iqtree.org/complex models, because I think I've put the wrong one. Here we go. Okay, I think what I want is this part. Let's just pop this in and make that a bit bigger. So the syntax is perhaps a little bit cryptic, but it's not too bad. We can say that that the actually this is, I use this one, Nexus format, because it's the same. Make that bigger. So this is how we can say the different character sets. That's char set. Part 1 contains these sites 1 to 100 and 250 to 384. Char set 2 contains, let's see, part 2 contains 101, 102, not 103, because that slash minus, it's like a -3, not the third codon. So 101, 102, not 103, 104, 105, not 106, and so on. That's the whole expression that does that. So 103, 102. So 101, 104, 107 from there, 102, 105, 108 from there. So that's how you can specify which parts you use in your model. And so because it's coding sequence, then you can say that part 1, part 2, part 3, however you want to describe them, can be evolving differently. An IQ-tree will allow those to have different models. So for example, that third position can be essentially much, much faster, or possibly even a different model from the first 2 positions. Here's another sort of a description of that idea. The character sets, part 1, part 2, part 3 here. And this is the partition of those character sets. And we can say that there's a particular model for part 1. This is called, it's a Goldman-Yang model, which is a codon model versus a DNA model. Don't need to worry about the names, it's just explaining the syntax. GTR plus G part model applies to part 2, which is these sites, 301 to 400. And WAG plus I plus G model applies to part 3, which is in fact that set of data, all the sites in that alignment. So you can specify all of those as much as you like in IQ-tree, and the complex model stock is really quite good at it, explaining what you need to do. So yeah, so for that I would say, since you've got codons, or at least it's coding, you can use a DNA model that accounts for that third position redundancy. You can specify it in not too horrible way, and allow that third position to be a different rate. You wouldn't change the first and second position to be different partitions, different in different partitions, that is to say, have different rates, because they're essentially the same, first and second. Third is the one you need to allow to be different. You also say that you're talking about looking at geographical separation, and it's within a species. So that may raises a couple of issues as well. Let's scroll down to here. So since you're looking within a species, you might want to find something that's evolving super, super fast. So you should check that the alignment, again, go back to your alignment, has enough information in there for you to resolve the phylogeny. Most programs, IQ3, Mega, Phylip, I think, does, RaxML does, they'll give you a count of how many so-called parsimony informative sites there are. Those are sites that differentiate in terms of their state between the taxa, so that they could correspond to, or they could support a branch in the tree. For a site to be parsimony informative, it needs to have at least 2 sequences that have the same characters as each other, but different from everything else. So you need to be able to say at least these 2 taxa are different from those 2 taxa. It might be these 5 taxa are different from those 4 taxa. That's parsimony informative. And you want quite a lot of those. Ideally, you want 100 or a few hundred. I understand that you've only got about 600 base pairs, so that might be a stretch, but do see that you've got some, or do look for how many parsimony informative sites you have. You should hope for enough to resolve all of the branches in your tree. There are some wonderful mathematical results to show that you actually need only a few parsimony informative sites in principle to resolve a tree, but those mathematical proofs are when your data are perfect, and none of us have perfect data. My phone is going off, but I have turned it off. Right. Now, since you're looking at geographical separation, you're also, you might be interested in the idea that you can make, well, you can make a tree from distances, and we've been talking about that with neighbor joining already. And so you could conceivably have a distance matrix that comes from real physical distances between your sites, your collection sites. And you could then see, if you build a neighbor joining tree using those physical distances, you might see whether it looks like your phylogenetic tree based on your sequences. Or you could see whether it's similar, whether it's perhaps statistically significantly close to your species tree or your subspecies tree. Because if it is, then you can conclude that there's some effect of either of geography on the species or species on the geography, which is unlikely. Another thing you can do is to simply plot distances, plot those distances against each other. This is a technique that's been around for a long time called independent contrasts. And what you do for that is you plot the sets of pairwise distances on the trees against each other. If you've got, say, taxa A and B collected from collection sites A and B, then, or 1 and 2, then you would look at compare the distance between A and B on the tree with the physical distance between collection sites 1 and 2, and plot the lot and see what the correlation coefficient was. You have to be careful doing that. It's not just you plot every pairwise distance in the geographical area and every pairwise distance with the on the phylogeny, because a lot of those distances are not independent of each other. They on the tree, the distance between 2 taxa, 2 leaves, could well be overlapping on that tree with another distance, because the distance on a tree is the sum of the path length, sum of the lengths of the edges of the path on that tree joining the two. I'm afraid I don't have a picture here. Whether I can make one at short notice, I don't think I can. But you can imagine if you're doing two distances or two sets of distances on a tree, and you trace through that tree to work out how far apart they are, those paths are often going to overlap, which means they're not independent. And if you're looking at something like the correlation coefficient for points on a graph, then you need them to be independent. What independent contrasts does is it finds sets of points on the trees that are definitely independent of each other because they don't overlap. So it's parts of the tree that don't overlap, and it plots those. If all of that is deeply unclear, then, as I've said before, just get in touch by email and we can nut out how that works. But it would be very interesting, I think, to see, for your question, whether the geography, the geographical separation actually corresponds to the phylogenetic distance. So for Cecilia from Monash, the question goes, I'm working on crop wild relatives and have generated phylogenetic trees for specific genes, which we have sequence for so far. I'd like to generate a multi-gene tree based on these as a consensus of the individual trees. So consensus is something we haven't really talked about yet, so it's a good opportunity to do it now. Oh, the last question is by Nilufar. Okay, I'm interested in your answers too. Okay, fine. Okay, so this one is, we've got multi-gene tree based on these as a consensus of the individual trees. Now, there's a couple of ways that you can create a consensus. You can concatenate all of the alignments and then build one tree from the alignments. But that's not what consensus methods do, what we commonly call them. Normally what you'll do in a consensus method is you'll build trees individually and then create a consensus from those trees. So the trees are now input to another process. There's a few different ways to do that. One of them is called strict consensus, and strict consensus is a method that just includes in the output tree, given the input trees, only the branches that are in every single input tree. So if there's any disagreement in the input trees, well, I should say it doesn't have to be in every single tree. It has to not be disagreed with by any trees. So strict consensus shows us in the resulting tree only the branches that are supported in all the input trees, or at least consistent with all of the input trees. So supported, I should add, or consistent with all the input trees. And that's your gene trees. Now, strict consensus is really strict, and it can easily be almost totally unresolved, if not completely unresolved, and you just get a star, which you probably don't want to see. And that can even be caused by a disagreement of just one taxon moving from one place in the tree to another place in the tree, because all of the branches are then not common. So or every branch in a potential tree is disagreed with by something. So that's not a great method. It's very, very strict. And those things can arise through those possibilities where you have the two different gene trees can arise with any of these processes that we've been talking about before. So horizontal gene transfer, incomplete lineage sorting, gene duplication, sampling error. All of those can give you a difference in your trees. Even though you've actually got the tree right, the tree doesn't reflect the species tree, so they can differ, even if you've estimated the tree correctly. That's the gene tree correctly. So strict consensus you can use, but expect disappointment. The next one would be majority rule consensus, and that's a consensus method that takes, if you have a disagreement between potential branches supported by their input trees, what happens is, if you have a branch that is in a strict majority of the input tree, so more than 50%, it can't be 50%, it has to be strictly more than 50%, then that branch gets put into the majority rule consensus tree. Because it's at least 50%, the strict majority part, that means that you'll be able to make a tree with all those branches. So that's actually quite useful. Majority rule consensus is a pretty useful consensus method. And I guess the last one, which is relatively commonly used, would be something called Adams consensus. This is a hard one to interpret. You have to be a little bit careful. The reason it's hard to interpret it is it's not actually showing you necessarily branches that are in any of the input trees or even in the correct species tree. The way it works is it puts each taxon, each leaf, in the most resolved position it can possibly be with respect to the root. So the furthest position from the root that it can be, as predicted by or as estimated by all of the input trees. So if you have some incongruence between your input trees, you might end up with an output tree from your Adams consensus that doesn't look like any of the input trees, doesn't necessarily agree with any of the input trees, but for each taxon in it, it will be the most resolved position, the furthest position it can be from the root in the output tree. It's a bit of a fiddle and it's a bit of an intellectual challenge to interpret them, so they're a bit less commonly used, but they can be pretty helpful. So if you are in the process of developing a consensus tree for your input trees, and again, do the lot, do all the different methods, see which one works, then it's a good one to include in that set. The other way I mentioned before of building a single tree from a consensus is from that concatenation of all the loci, and I think we've discussed that pretty well by now. So with any luck, you'll have a sense of how to do that. At least have at a minimum allow different models for each locus. But I actually don't think that's the question that you're asking here. One of the fathers of phylogenetics is Joe Felsenstein, and I think although he's retired now, he still has a list of phylogenetic software which is maintained. It's available through this website, evolution.genetics.washington.edu. Phylip is a suite of programs that he wrote in C. Is it open source? I suppose it is open source. You can get the code, compile it, and run it. They're still very efficient. Doc is for the documents, and consents.html gives you some details on the program also called Consents, and that makes consensus trees based on input trees. It's not the prettiest of interfaces, but it does work very well. And there are some other methods which look prettier, and I think they essentially do the same thing. So one other is called Dendroscope, and there's a site, a URL given here, which you might need to paste into a web browser. I think it's put an accidental space in there, so just be a bit careful posting that in. So that does some consensus methods. So does Mega. As I said, I don't use Mega, so I haven't got experience in using that for consensus. Splitstree also does consensus methods, and this raises another level of complexity, I guess. Version 4 of Splitstree, and that's the one that we used, only has the first two consensus methods that I mentioned, the strict and the majority rule. But version 5 also has the idea of a consensus network. So it takes input trees and produces a network from those trees. And that can be actually quite informative as well. I hope that answers your question. I really do. There are a lot of different ways to build consensus trees. You would hope that the individual loci, individual genes, will give you something similar. They should agree roughly on the phylogenies of your crops. Where they differ, then it might be down to probably not HGT. It might be hybridization. It might be incomplete lineage sorting or sampling error. But that should come out when you compare the trees that you have. If you have lots and lots of trees, like hundreds of loci, it gets pretty hard to compare them, but you should also be able to find software that will identify which trees are the same. So if they are differing, then even if you have lots of loci, most of them should be, one type or another type or another type. There will be several genes that give you the same tree, we would hope. If everything gives you a different tree, then there's something very funny going on indeed. This is another question. This is a little fast question. I've been working on multi-locus phylogeny of Cercospora. Actually, I had to look this one up. It's a fungal genus using 8 loci. It's a very species-rich genus with hundreds of known species. Depending on what species I include in the multi-locus tree, sometimes I get quite different topologies for some clades. Any ideas on how I should proceed? Well, I think if you've got 8 loci, you should build a phylogeny from each locus that you have. And if you have masses and masses of species in your analysis, it might be hard to compare the trees that you get from those 8 loci. But I would try that first. I know that it's tricky to visualize hundreds of species in a tree, but it is possible to print the whole lot out, get out with a highlighter, and I know because I was doing it last week, and see what the differences are between those 2 trees. In your case, 8 trees. So I would hope that you could get the 8 loci and get your phylogeny right for each locus, because there's very likely to be some HGT, or maybe, I don't know whether fungi can hybridize. Maybe they can, I honestly don't know, but there might well be some lateral gene transfer going on, which could give rise to some different phylogenies there. So I would say yes, it's probably quite likely that you'll get different topologies for different loci on the same clade. At that point, we can, or you can, if you'd like to, you can ask me offline as well. You can have a look at what the differences are across those clades. Maybe if they're statistically significantly different sub-trees, trees for those clades, maybe you've got some HGT going on. But yeah, basically do each locus separately and look and eyeball it, and load them into I didn't even finish that sentence, a consensus or tree display program. Okay, tree display. I'm going to take a little break for my throat, and if anybody wants to put some more stuff into the discussion board, then we can come back and answer those questions in, say, 5 minutes. Okay, all right, so let's pick up. There's been a couple of questions. I'm going to just copy paste them into this document since I'm sharing that. makes it a bit easier. Everybody can see it. So I guess the follow-up ones, thanks for the positive feedback. Right, so yes, fungi can hybridize. Great. So that's, yeah, terrific. That makes everything pretty hard. So I'll just copy that question into here. I've made a note for myself to maybe come back to that. So the question is, yes, fungi can hybridize. Do you mean that I can combine the 8 trees from 8 loci after I built them? Yes, I do. You can do that. Bigger. But what I would do, in fact, is look at the trees that you get from the 8 loci. I'm not sure how many taxa you're talking about. So if you can put that into the document, that would be helpful. If it's just a few, it's actually quite easy to visualize to see the differences between them. If it's a lot, then it makes it harder. I will say that there is a nice bit of a consensus method that you can use to construct a consensus network based on input trees. So if you have 8 input trees and they're different, but they don't all have the same taxa in them, you can still use this consensus method. It's called Z-closure. Z-closure. And I think that is the method that is used in splits tree. So if that's correct, you might have to check, but just off the top of my head, I think that's the one that's used in splits tree. That's neat because you don't need all the taxa in every tree, which is pretty helpful. Okay, so between 30 and 40 species there has been the answer to that. I think this is something that you could simply print off 8 trees, 8 trees, and on 30 to 40 loci and compare them by eye. If they're similar, great. If they're different, then it's something that you can get a consensus network for, or a consensus tree if you have every sequence covered. If that's a bit of a challenge, if it's not working, then drop me a line and I can help you with that. I've got another comment on there. I think that's come. I think that's something you can evaluate with Astral. Oh, okay, yes, Astral. I can't remember about Astral. So that's on the discussion board there. XVC has suggested that can be checked with the Astral program, and there's a URL for that. Okay, that's cool. And part of the question was, should you choose congruent loci only? No, not at all. They're all telling interesting stuff. Provided you've got sensible data in each of them, then include them all and compare your trees that way. You might find that there's HGT going on, which would be interesting. Okay. Another question is from Cecilia, and I'll just pop that one into the document in front of me and make it a bit, okay. So there's no species tree available for these wild sorghum species yet. And that was, what was Cecilia's question? Sorry, I'm scrolling up. I don't want to disorient anybody, but that was on the crop wild relatives, phylogenetic trees for specific genes for which we have sequence, wanting to generate a multi-gene tree based on those as a consensus. All right, so let's pop down to answering that. No species tree available yet for these wild sorghum species. So if you've got, and what you have now, and I remember we discussed this a little bit last time, is that most methods infer unrooted trees. And if you're after a rooted tree, you do need something like an outgroup. I've got a bit of feedback. Someone's unmuted. So yes, the suggestion that you should get a homologous gene from the closest species to root the tree, that is spot on. That's what you need. Bear in mind earlier comments about, excuse me a sec. Earlier comments about balancing the sort of lengths of the branches of the tree. If you have the closest species is joined to your in-group, that's the species of interest, by a long branch, you might want to actually collect a few closest species, relatively close to the wild sorghums, but you know that they're not wild sorghum. So they're outgroups, but if it's a group of them, you might be able to break a long branch into a few shorter branches. The problem with that long branch coming into a closely related group of species is that it's very hard to pinpoint exactly where a long branch should go, whereas if you've got a bunch of sort of shorter branches, it's much easier. Statistically, it's more likely to get the position of the root correct. Okay, so I'm looking at 20 genes across 6 wild and 1 domestic, so 7 species. I would hope that you've got, that's right, 8 trees, yes, okay. So I'm confusing myself with the question above, so I'm going to just make myself not see that anymore. So there's no species tree available yet. I think what you're after is a consensus. So if you've got an outgroup for your in-groups, your wild types and your domestics, your 7 species, you need 1 or 2 maybe closely related taxa for an outgroup. And then I think what you need is a consensus method to at least begin to show what the species tree is most likely to be. Once you've got that, and for only 7 species, it shouldn't be too hard to get right. Once you've got that, you can then go back to your individual gene trees and perhaps map them into the guess at the species tree and see which ones deviate a lot. Another thing you can do is this consensus method using Z-closure. If, as I've said, that's the method in splits tree, it'll allow you to make a consensus network, and so you can get a sense of what the conflicts are in your trees. It doesn't tell you that those conflicts are due to HGTs, horizontal gene transfers. They might be, but they might also be those other sorts of things that can give rise to conflicting phylogenies, like incomplete lineage sorting or sampling error. So it sounds like you've got quite a good data set. If you've got 20 genes and you've got reasonably good sequence, then you should, without too much difficulty, be able to, again, print those trees and eyeball the differences. If they are very, very different from each other, then that consensus network or a consensus tree will be very badly resolved. And so you'll also be able to learn that something has gone on in these relationships between these 20 genes. You should also be able to do, just because you only have 7 species plus a couple of outgroups, you should be able to identify which genes are in agreement with each other, hopefully all, or which genes are disagreeing with each other. So I think that would be how I would tackle that one. And I would use, because I've got genes, I'd be using a likelihood analysis, let IQ-Tree pick a best fit model, and do bootstrapping. Even though it doesn't give you confidence, it gives you a sense of how consistent your data are for each gene. And you should get a sense of how robust each of those gene trees is in your data set, your 20 genes. I hope that helps. Okay. Right. Now I'm actually at the end of all the questions, and I think, yes, people are still typing. So Oh, that's a thank you. You're very welcome. I think if there are, we'll leave it a couple of minutes, and if there's no more questions, then we'll call that a day.
All right, so thank you very much, Michael, for your time and expertise in helping people with their individual questions today. It's been very valuable to those people whose research problems you've addressed. I'd also like to acknowledge the University of Tasmania for generously contributing your time and efforts to these two workshops. They've been fantastic resources that will continue to be useful via the recordings on the Australian Biocommons YouTube channel. We've mentioned a few times during the course of these two workshops that there will be a follow-up event. If you'd like to hear about that in the future, please make sure that you are in touch with us via a couple of ways. You can follow us on Twitter at AusBiocommons, and you can also subscribe to our newsletter at the site you can see on your screen. If you'd like to go back and revisit these sessions or previous training content, you can do that on the Australian Biocommons YouTube channel, and we will have a regular webinar series coming up next year, as long as more hybrid training events. Now, I'd just like to finally finish off the session by acknowledging our funders. The MBAL Australia Bioinformatics Resource and Australian Biocommons have both been supported by Bioplatforms Australia, with the Australian Government investment through the NCRIS program. So once again, thank you everyone for coming along to this session and to part A, and thanks once again to Michael Charleston from the University of Tasmania. Goodbye.
Bye, everybody.